Now, I'd like to invite Arielle Lofchick, who will be addressing us today, giving her talk, yeah, assessing climate change impacts on pollinators and forage plants in Mediterranean shrublands.
So thank you so much for having me here. My name is Arielle, and I study with Avi actually in the Hebrew University of Jerusalem. And I'm very excited to present my preliminary results from my first year of my PhD. So I will begin with the buzzword climate change. Sydney already did a great job explaining the change in weather patterns and plant phenology, but for the sake of my presentation, I will continue. So in general, scientists agree that precipitation patterns will alter seasonally, annually, and all around the world. And experiments are urgently needed to support or disprove widely used models that predict future ecosystem properties based on current climate conditions. On the impact on pollinators, sorry, first, with climate change, we can see a decrease in colonization and declining populations and pollinators, and even extinctions in regions over time. And moreover, as Sydney did an amazing job explaining, is there can be phenological mismatches where plants and pollinators are no longer in sync with their timing. Additionally, it can alter floral resources and even floral phenotypes, affecting the interactions between plants and pollinators. And while these changes can be subtle, this may affect how they interact with flowers, their visitation behavior, and even how they handle the flowers. Ultimately, these changes impact pollen transfer for plants and foraging efficiency for pollinators. Now, I'll talk about the effects of water deficit on plants. We know that increasing stress in plants like drought can affect floral development, including floral morphology and phenology, as well as nectar and pollen production. Flower morphology refers to the form and the structure of flowers, including the arrangement of the petals, the sepals, the stamens, and pistils, which are critical for reproduction and species identification. Now, my experiment takes place in the Eastern Mediterranean basin in Israel, and if you follow this map, you will see exactly where it is. And the Eastern Mediterranean basin is a biodiversity hotspot where we have very hot and very dry summers, and we have wet winters, approximately 100 to 600 millimeters of rain in the winter. And scientists agree that we will see a decrease in precipitation and an increase in temperature. Now, specifically, the name of my research site is the Meta Long-Term Ecological Research Station. And here we use we use rainout shelters to manipulate the rainfall amount and frequency of rain. So, you will see that we have 4 treatments. We have the high treatment. You can see here the rainout shelter. And in the high treatment, we have a 66% reduction in rainfall, and it's irrigated 9 times per year in the winter months. And we have the same treatment, which is the low treatment, which is also a 66% reduction in rainfall, but rather than a 9-time irrigation, it's only irrigated 3 times. And the irrigations are very far apart. We also have a natural frequency treatment. So this treatment receives natural rainfall, but as you can see the stripes in the middle, they collect 33% of the rainfall. 66% of the rainfall, so only 33% of the rain gets in. And so there is no change in frequency in the amount of rain, in the frequency of rain. And we have the control treatment, which is a natural precipitation regime. And this is just a nice photo overview of the experimental setup. So we have 4 treatments, the high frequency, the low frequency, the natural, and the control. And we have 5 repetitions in blocks. Now, the goals of the study were to study the effects of drought, both the amount of rainfall and the frequency of rainfall, and how this affects foraging resources, pollinator preference, and activity. And we chose to focus on the plant Salvia fruticosa, which is also known as Greek sage. It's very abundant in this area, and pollinators really like this plant, and it's also very easy to work with. In this experiment, we measured many variables, but because of the time limit today, I'll only talk to you about the flower phenology of the Greek sage. So we measured the beginning of the bloom to the end of the bloom in all four treatments, and also the flower longevity. So as you can see, a bud here, we measured the bud from bud to all of the sexual stages to the fall of the flower. We also measured the nectar volume in the Greek stage, and we also did plot level visitation observation. So I'll quickly show you my preliminary results of what I found so far. Sorry. Here we see the mean first bloom date of the Salvia fruticosa. So we have the treatments on the y-axis and the date on the x-axis, and you can see that the control treatment is blooming significantly earlier than all three treatments, the natural, the low, and the high. And it's very significantly different in the low treatment, which is a 66% reduction in rainfall and only 3 irrigation frequencies. Now, when we look at the individual flower where we monitor the lifespan of the flower, we see that the high in the high treatments is 66% reduction in irrigation and 9 irrigations. This flower is actually living longer than the control treatment. And we did not find any significant difference between the low treatment and the natural treatment in comparison to the control. When we look at the nectar volume of the Greek stage, we decided to focus on the female stage of the flower because this is the most important part for pollination when the stigma is receptive. And we can see that there was a significant difference between the high treatment and the natural treatment when we compared nectar volume. Now we compared nectar standing crops. So what we did was we covered the flowers for we drained the flowers and then covered the flowers for 24 hours. And this is a representative volume of what nectar is available to the pollinators. So it is interesting to see that we're seeing higher nectar volume in the control than the other two treatments, high and natural. And finally, when I did pollinator observation, I saw no significant difference between the 4 treatments. So what I did was I did 10-minute observation transect. So I walked around the entire plot and recorded what I saw. And I didn't see any difference between the treatments. But there's one important thing to note is that all of the plots are very close together. So we need to consider the species within the plots and also the behavior moving forward. And so my preliminary evidence shows that the frequency and amount of rainfall is affecting the flower phenology and the nectar production, but we're not seeing any effect on pollinator activity yet. So I'm really looking forward to the field season next year to continue the research. And I want to say thank you to my university, both Tel Aviv University and the Hebrew University of Jerusalem, the Faculty of Agriculture, and the Jewish National Fund for their funding.